Hello everyone, welcome to Miss Doc T. This is Dr. Farheen, General Medicine Resident, and today let's discuss iron metabolism. Iron is absorbed from the proximal duodenum, and in the food it is mainly in the form of ferric iron. But to cross the duodenal membrane, iron should be in a ferrous form, and so the ferric iron gets converted to ferrous form by an enzyme ferroreductase on the luminal side of the enterocyte. The ferrous iron then enters the enterocyte of duodenum through divalent metal transporter 1, and then the iron reaches out of the enterocyte through another channel known as ferroportin. Now the iron here is in ferrous state and it is to be converted back to ferric state with the help of ferrooxidase on the basolateral side of the cell membrane. Ferroreductase is also known as duodenal cytochrome B, and ferrooxidase is also known as hephaestin. So hephaestin mediates the conversion of ferrous iron to ferric iron, and duodenal cytochrome B mediates the conversion of ferric iron to ferrous iron. Coming back to the iron metabolism, the ferric iron is then complexed to transferrin, which is shown in green, and then it circulates in the blood till it reaches the two main target organs having the transferrin receptors, that is the bone marrow and the liver. Now let's see what happens to the iron reaching the bone marrow. The transferrin bound iron attaches to the transferrin receptor, and the whole complex is then endocytosed into clathrin coated pits. Then the iron is taken into the bone marrow and rest of the complex is released back onto the surface. The transferrin receptor gets attached to the cell membrane and the transferrin is released back into the circulation. The iron in the bone marrow is utilized for erythropoiesis, and so the iron gets trapped in RBCs. After 120 days, as the senile RBCs reach the spleen, RBCs are broken down in the splenic macrophages and the iron is again released back into the circulation through a channel ferroportin. This released iron again binds to transferrin and it circulates in blood. So bone marrow is the first receiving site of iron, and the second organ is liver. Liver takes up iron through the transferrin receptor and it mostly stores it in the form of ferritin. And whenever required, iron is released from the liver through the channel ferroportin. So ferric iron is absorbed from the duodenum, is converted by the duodenal cytochrome B to ferrous form, and then through the divalent metal transporter 1 it enters the enterocyte, and through the ferroportin it leaves the enterocyte and it is converted back to the ferric form by hephaestin or ferrooxidase. Iron binds to transferrin and circulates till it reaches the target organs having transferrin receptors. The first is the bone marrow, where the iron is utilized for erythropoiesis and it is trapped in RBCs, and after 120 days when the senile RBCs are broken in the splenic macrophages, the iron is released back into the circulation. And the second organ with transferrin receptors is the liver. The iron entering liver is mostly stored in the form of ferritin and it is released into circulation whenever required. So one important point to remember here is that there is no active excretion of iron, and iron is lost only passively through shredded epithelial cells or through blood loss, and so absorption and iron metabolism should be tightly regulated to prevent iron overload. Iron is utilized mainly by the bone marrow and the liver, and iron reaches circulation from three sources. First is duodenal absorption of dietary iron, release from the splenic macrophages and release from liver, and all the three require the same channel, that is ferroportin. So this ferroportin is tightly regulated by a hormone released from the liver called the hepcidin. And hepcidin is a very important regulator of iron metabolism. Hepcidin is also an acute phase reactant, and so in inflammation, especially mediated through interleukin 6 and some bacterial polysaccharides, can increase the hepcidin levels. So this increased hepcidin inhibits ferroportin, thereby decreasing the iron levels in the body. On the other hand, abnormalities of hepcidin gene or the HFE gene decreases the level of hepcidin and so decreases inhibition over ferroportin, and so iron is increased in the body, leading to iron overload and hemochromatosis. So increased hepcidin inhibits ferroportin and iron decreases in the body, leading to anemia, which is known as anemia of chronic diseases or anemia of inflammation. And decrease in hepcidin decreases the inhibition of ferroportin, so more iron enters the body, leading to iron overload and hemochromatosis. Just a simplified presentation of role of hepcidin. Iron is absorbed from the duodenum through ferroportin. Iron is released from the splenic macrophages and from the liver into circulation through ferroportin. And increased hepcidin inhibits ferroportin, so iron decreases in the body. And when hepcidin is decreased, there is no inhibition of ferroportin, so iron increases, leading to hemochromatosis. So this is the complete iron metabolism. Some points to remember here. Iron to cross the duodenal membrane should be in the ferrous state. Iron circulating, that is bound to transferrin, is in ferric state. The storage form of iron, that is ferritin, is in ferric state. Hepcidin is the major regulator of iron metabolism as it regulates the channel ferroportin. So decrease in hepcidin increases iron levels leading to hemochromatosis, and increase in hepcidin decreases iron levels leading to anemia of inflammation, also known as anemia of chronic diseases. Not to confuse hepcidin with hephaestin. Hephaestin is the ferrooxidase enzyme which is present in the basal side of the enterocyte membrane that helps to convert ferrous iron to ferric form. So this is all about iron metabolism. And that's all for today. Thanks for watching. Hope you liked it. Let's meet again with another new topic. Till then, just take care, be happy, and stay safe.